All right, guys, so this is your first set of lecture notes. We're going to start with just science and scientific processes. So obviously you are in advanced biology here at Hoover High School as a freshman. In general, we tend to talk about biological sciences, chemical sciences, and physical sciences, kind of as your three main disciplines. And then there's some characteristics in general of science and scientists that we're going to hit kind of quickly here. You've probably just got some basic understanding of this from middle school, or just it's kind of common sense. So as scientists, and I am a scientist by degree, I have a bachelor's in zoology and a master's in biology. My bachelor's is from Auburn and my master's is from UNC Wilmington. But in general, scientists, we tend to be curious, okay? Asking questions, we make observations, we want to understand processes that occur in nature. And nature here is going to mean biology, chemistry, and physics as far as natural phenomenon, okay? This is not just like trees and rabbits, okay? So these are things, disciplines that are studying nature, right? So science, we got to be organized to some extent. Some scientists are kind of a hot mess, but organization is important. Scientists tend to be very data-driven, be interested. Ooh, sorry, I just kind of made that fuzzy. So let me refocus for a second. So doing research, predicting, creating, analyzing, making inferences. I've got the definition better written so that I can see it on the next slide for that. So that's why we're at definition coming there. In general, it used to be that we would say, here's the scientific method. Well, there's not a one lockstep procedure for every experiment. So now we're just going to talk scientific processing concepts. So that's, again, more to come. Scientists in general, we tend to understand that science changes, that we learn new things all the time about what's going on in the world around us from a natural standpoint. Again, we're talking biology, chemistry, physics, earth science, space science. Science changes, okay? And that's part of just the inherent nature of the world and how the world works. Okay, so let's go to the next slide. All right, so scientists decide what to study in a lot of ways, which I know you can't read this at all on my lecture notes here, so I hope you have your own out. Oh, and I just realized, I think I forgot to say at the beginning of this, if you have a question, write it down. If I tell you to write something down to enhance your notes, do so. Or if you just hear me say something as an example or something that's not on here, then I would write it down as well. So how do we decide what to study? Well, obviously doing some research. That's the first thing that says. Does the person have general interest about a topic? And then the next couple, lab space, cost, and travel, those three. So do you have space to work? Do you have materials that you need? What's the cost of doing this particular research? Do you have to travel? And travel costs money. So just some kind of logistical things there. Does a scientist have prior knowledge and experience on some particular subject? Can they get test subjects? So you want to do some study where you actually need human subjects. Can you actually get them to participate? Trial and error is really important in science because sometimes scientists are doing some particular experiment, conducting it, and then they get enough information from that experiment, but then there's some major error that happens, and so as a result it kind of gives them an offshoot to jump into a new experiment. And then just, I don't know, you can probably, maybe you can think of something else that might determine how a scientist figures out what they want to study. The next bullet with what does research involve, obviously for us in this day and age, our first go-to is going to obviously be the internet. But I've got books, interviews, observations about the world around you, inferences, which I'll come back to that with just this definition in just a second, magazines, TV, etc. Okay, and then research, that second bullet right there says that it can be done during and after scientific experimentation. And then in general, research these questions over here, who, what, when, where, why, and how. Those are going to be your basic questions to just get you started on learning any new topic. So that's a good way to just start from a scientist standpoint as well. Let's talk about inferences for a second. So inferences, by definition, they are logical interpretations based on prior knowledge. And it says that on your sheet there, but it's in parentheses. And of course on mine you can see that if this were on the screen, we would be able to see that pink much more clearly than we can here. But that's just going to be how it is because I printed this already. So let me give you an example of an inference in my room. So hopefully you're going to remember to look at this when you come back in here. So if you look up at the ceiling, if you're sitting at the tables facing the front of the room, if you look to the left, it's way worse than it is over to the right. But on the ceiling at the left, you can see that there are some brown stains. And I always use this as an example of an inference. So what might that be? Well, when you come back in here, look at them, and you'll probably realize that they are water stains. Maybe the roof leaked, maybe there's a pipe that burst, and they've been here as long as I've been here. And this is me starting my 20th year here at Hoover High School. Another example, let's say you pull up in the parking lot at Publix, Walmart, Target, wherever you're going, and you smell fumes. You can probably say to yourself, oh, I bet somebody was just parked here a few minutes ago, like even if you didn't see the car pull out or something, but you smell it, you know there was a car there. So those are just a couple of examples of inferences, and you'll be asked to make inferences. For any experiment, essentially to get started, the best thing for a scientist to do is determine what they want to study and put it in the form of a problem that they want to solve. For us here at Hoover High School in advanced biology, you're going to use this problem statement, and it says the effect of the independent variable on the dependent variable. So if I ask you for a problem statement in the correct format, it always goes in that form, or it's going to automatically be marked wrong. That being said, let's give an example real quickly. So let's pretend that I said, all right, we're going to study the effect of light intensity on the growth of plants. And I gave you your group 4 plants. One had to go in the closet in the dark that's in my room, one under a 25 watt bulb, one under a 75 watt bulb, and one under a 100 watt bulb. And you had to measure the height in centimeters. So your independent variable would be the light intensity, because that's what you're changing. Your dependent variable would be the, I just lost my train of thought. Your dependent variable would be the height in centimeters. All right, let me shift gears. Sorry, you just saw my hand there. I could have paused it, but chose not to. Okay, so next, hypothesis. So we're going to write hypotheses in the same format all the time in this class as well, that if-then statement that you can see there. So a hypothesis, by definition, it's a scientific explanation for a set of observations that can be tested. It says tests allow for data to either support or reject the hypothesis after completion. So your hypothesis cannot be right or wrong, and I would write that down, okay? It's an educated guess. Sometimes we hear that phrase, but that's not the actual definition. So you can't say my hypothesis was correct or incorrect. You can say the data either supported the hypothesis or it rejected the hypothesis. Okay, or you could say the hypothesis was accepted or not accepted. That's another way to talk about that. So for us, and right here it says every time, so I'm going to say the same thing here. Use this every time when you're writing hypothesis in this class. So it says if the independent variable is, and then that little set of parentheses says describe how the independent variable changes, then the dependent variable will, and then you predict the direction of the effect. So let's go back to our plant example. So if the light intensity is, I'm going to say increased, then the height of the plant will increase. Okay, and that would be a good good hypothesis for that particular experiment. Okay, let's say that you think that too much light is going to prevent the plant from growing a lot. So you kind of think, ooh, under that 100 watt bulb, I think that's going to be a problem. So you might really just say, if the amount of light decreases, then the height of the plant will decrease. It's probably going to be that based on prior knowledge, your inference would be if the amount of light increases, then the height of the plant will increase, because most people in the 9th grade know that light impacts plant growth in a positive way typically, okay, and causes plants to grow. So this next section, it says carefully designed and controlled experiments will test the hypothesis. Again, data will either support or reject the hypothesis. So you're going to have to deal with a materials list. What are the materials you're going to use? How are you going to reuse them? Writing up procedures, all kinds of things involved there, okay? Data then at the end, once you've collected it, it's recorded, organized, and analyzed, and then conclusions are drawn. Trials will be repeated if need be. Obviously in here, we can't do repeated trials like as a group. When you are all carrying out a particular experiment within class, sorry, I needed to focus that again, I felt like. When you're carrying out an experiment in class and say we've got a handful of different groups, that becomes our repeated trials, okay? But we draw conclusions, we repeat trials, and then we also deal with sources of error, which that's coming in another slide, okay? So, that being said, what are the parts of our controlled experiment? Okay, so let me slide this up. All right, so the first thing is the control or the control group. So this is the part of the experiment where the factor or the key factor that's being tested doesn't change. We often hear this phrase, standard of comparison, okay? So it's what you're going to compare all your other groups to. And your other groups are called your experimental groups. Okay, so that's something I would say you need to write down, okay? So again, this is either the control or the control group. So in our plant example, this might be the plant in the dark, in the closet. It's getting no light, okay? So then that means that any change in light intensity should affect plant growth. Your 3 plants under the light bulbs, and what did I say, 25, 75, and 100 watts, I think. Those 3 groups would be your experimental groups. So you can have as many experimental groups as you want in an experiment. You could have just the 3 in our plant experiment. You could have 100. You could have 1,000, right? Just depends on what you're testing. Let's see. So that, again, that's all your other groups. So then number 3, the independent or the manipulated variable. This is the term, I'm going to do this for both this and this, okay? Independent and dependent. Manipulated and responding are just ways to think about this, and we used to have a textbook that used those terms, and I like explaining it that way, but that is not what you're going to see when I'm asking you questions. So the independent variable is the variable that is being changed or manipulated. It's what the scientist is studying or testing. And so again, for us, that's going to be our light intensity in the example that I gave you a little bit ago. The dependent variable, this is what you're going to measure. Okay, this is what you're collecting data on essentially. This is going to respond to the change in the independent variable. Okay, so again, we're going to use independent and dependent. So in our plant example, this would be the height of the plant, right? That's what we would measure. And it responds to the change in the light intensity. Okay, so then constants, factors that are kept the same in all groups in an experiment. So we want to make sure we're holding lots of things constant in our plant example. And if you were in class with me, I would ask you for some specific examples here, and I'm sure you could give me some great ones. Okay, so I'll give you a few, and then I want you to think of, are there some others? So we would want the same size pot, okay, for our plants. We would want to use the same type of plant. We would want to use the same type of soil. We would want to have the plants exposed to the same amount of, or sorry, not same amount of light, but to expose to the light for the same amount of time. Say that we turned the lights on. I turned them on every morning when I got here, and I turned them off every afternoon when I left, just strictly to, you know, not have them running all the time. If we were worried about a fire starting or something like that, we would want that to be the same. Or maybe I left them on all the time, and then we ran the experiment for a week for every single plant. And then that plant in the closet has to stay in the closet for the same amount of time. Okay, I bet you can think of some other constants, right? why don't you write down another constant or two that are past the ones that I just listed? All right, so this last term you may not have ever heard. This is the operational definition. So this, by definition, describes how a particular variable is to be measured or how a term is to be defined. And this says right here, how do you define cold? Okay, so that being said, would we define this just kind of qualitatively, like it's cold because I need to put on a jacket, or it's not cold because I don't feel I need a jacket? Or do we actually use a thermometer? Okay, pretty simple. For our plant experiment, your operational definition could be the dependent variables, the height of the plant. The operational definition would be the height in centimeters. And we would use centimeters, let's say, for a small plant because we are in science and we use the metric system. Okay, say it's a huge plant, then you would maybe measure the height of the plant in meters instead. Okay, so your operational definition is basically units, okay, description. Like I said, is it cold enough to need a jacket or not? That's more of a descriptive operational definition. Okay, units are often, oh, you can't see that, sorry. Units are often your operational definition. Okay, next. All right, so observations. In general, we make observations using our 5 senses. Oh my gosh, I am not moving that the right direction, am I? Okay, so observations are made using the 5 senses. So we gather data that way. So I would list your 5 senses on your piece of paper. If you can't think of them, okay, then look them up, okay? I'm not going to tell you what they are. Right, so we would gather data through observations. Data, by definition, is simply just information or evidence. Data can be either quantitative or qualitative. Okay, quantity means number, so that's your numeric data. Again, we're going to use the metric system in science. So how many? Just are you counting something? Okay, there's 5 people, 10 people, 20 people. How fast? Okay, is your car going 50 miles an hour or 80 miles an hour? What percentage? Okay, what percent of our student body is male versus female? Right, then qualitative. This is descriptive data. So color, shape, scent, texture. So wherever you are right now, kind of look around and see, can you collect real quickly some quantitative data? Okay, just looking, using your eyeballs in your room or your kitchen, wherever you are. And then qualitative data. Can you do the same? Can you collect some quick qualitative data? The color of something, the shape, how does it smell? Texture, is it smooth or is it rough? Can you tell your mom's baking cookies, whatever it happens to be, okay, because you can smell them. All right, and so then data is organized into tables and graphs. Okay, so this is a table with rows and columns. Okay, this is a bar graph. And this is a line graph. All right, so you're going to have some notes on graphing as well, and do some graphing for me pretty quickly, and you need to read those notes. Basically, as far as all I'm going to say right here about this is we've got to organize our data into tables, and then from tables we make graphs, whether it's continuous data that changes in some capacity or it's data that's comparison data, okay? Your line graph would be, whoops, wrong direction yet again, sorry y'all. Your line graph would be for data that's going to change over some sort of interval, and again, bar graph to compare. Okay? So this might be in Mrs. Zellmer's classes, how many people prefer like chocolate candy in blue versus like sweet tarts, nerds, sour straws, things like that, maybe in green. I don't know. Okay, so then once it's organized, then you can make your comparisons, you can interpret the data, you can analyze the data, and then notice here it says too that inferences are sometimes made during data analysis. So sometimes you go, I wonder if that data is because of this, and you base that thought off of something you already know. Okay, so again, this is in we're going to compare, we're going to interpret, and we're going to analyze. conclusions are drawn. Okay, so this last slide of the science portion of this, conclusions are drawn after that data is collected, and you're going to basically state either the data supports the hypothesis or rejects the hypothesis. Notice this says supports or contradicts. You don't say, again, that the hypothesis was right or wrong. So here's a good example of how if I said to draw conclusions and to state them, the hypothesis was not supported because the data shows, and then you finish your thought. Okay, one of the things you're going to be asked to do is explain sources of error, whether it's human error or error with equipment. Okay, like let's say one of our bulbs burnt out and we didn't know it. Well, that might impact the height of our plant that was sitting under that bulb. Okay, then you're going to be asked for possible extensions or modifications to the experiment. How could you take the exact same experiment and just extend it, do a little bit more, learn a little bit more? Or it could be a major modification, okay, or a slight modification. So for our plant one, maybe we just change the type of plant, okay? Maybe we decide to, what was I going to say? I just lost my train of thought again. That we change the type of plant, or that we decide to measure after 2 weeks instead of 1 week, okay? Or we measure every other day instead of every day. It could be something really tiny, right? Or it could be something really major. And then it says valid experiments must be tested many times. So again, many test subjects, multiple trials, okay? And it just kind of depends on what you're doing as to what the need is there. Okay, and that is it on just basic science and scientific processing. You will have, again, some notes on tables and graphs that are separate that I don't think I'm going to lecture on per se, so you'll need to ask some questions about that if need be.